Hello and welcome to part two of the summer lecture series. So today we are going to be talking about basic chemistry stuff. So we'll be going through this rather quickly with a few tangents that will definitely relate to things that we talk about later. So basic chemistry, this is going to be easy stuff. All right, our basic chemicals that are going to be important in biology are going to be the sponge elements. The sponge elements are going to be sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. These are the main ones that make up the bulk of your body. Of those six, oxygen, carbon, and hydrogen will be the highest in your body, followed by nitrogen, then phosphorus, and last sulfur. But there are about 25 elements in total. Other than these main six, you have trace elements that are going to be important to make things like enzymes function. So you need things like copper, you need zinc, but only in very minute quantities. So you don't need to go eating pennies or eating nails. You also need things like iron for hemoglobin. You also need iodine for making certain hormones in your thyroid. So if you don't have these trace elements, then you'll begin to have different metabolic issues affecting the way that you process food, the way that your enzymes work. And in the case of iodine, you can get a goiter. Okay, so what we see here are two examples of nutrient deficiency. Over here, we see that we have corn. We have this corn right here that is nice and tall and very dark green, and this corn over here, which is a lighter green and a little bit shorter. That's because the corn here has plenty of nitrogen, and therefore it can make more chlorophyll, which can allow it to photosynthesize more and therefore grow bigger. Over here, there's nitrogen deficiency, probably because in previous years this set of land was planted with corn before, and corn is a big nitrogen hog, so it sucks that nitrogen out of the soil, depleting it. And then this side over here was probably treated with some inorganic fertilizer, helping it to grow better. Problem with inorganic fertilizers, they will run off into waterways, causing lots of algae to grow, and it will also affect soil quality if it is an inorganic fertilizer rather than an organic fertilizer. We'll talk about that stuff later. Now this woman on the right, you can see this large lump right here. She did not swallow a potato. That is her thyroid gland, and that is what's known as a goiter because of an iodine deficiency. So the way that the thyroid works is it makes two main hormones, triiodothyronine, that is known as T3, and thyroxine, which is known as T4. And it's 3 and 4 based on the number of iodine atoms that are in them. So if the body does not have enough iodine, it cannot make these two hormones. Hormones are responsible for things like metabolism. They affect heart rate, they affect blood pressure, they do lots of different things. If your T3 and T4 hormones are low, then the brain sends a signal called thyroid stimulating hormone, which we'll just call TSH, thyroid stimulating hormone. And that tells the thyroid to make these two hormones. But without iodine, the thyroid can't. And so the brain keeps pumping out thyroid stimulating hormone, saying, make the hormones, make the hormones, make the hormones. And of course, the thyroid can't. So those constant signals cause the thyroid to become inflamed and grow, and that's why you get potato neck or a goiter. So if this woman were to take some iodine, that would eventually go away. We typically do not see this in first world countries like America or in Europe because we have plenty of iodine in our diet, because it is in table salt. They add a potassium iodide into table salt. Table salt is sodium chloride. And they do that so that we have enough iodine in our diet to keep our thyroids healthy. You can also get iodine by eating kelp. Kelp is a seaweed, and it is used in lots of different kinds of Asian cooking. It's also in other types of seaweed, such as the stuff around sushi. So if you're eating sushi, you are getting iodine. All right, basic composition of a human body, symbols of elements, and basically what we're made of. We are mostly oxygen, and then carbon, and then the other things. Then down here are your different trace elements, so you can see all these different things that we need. And most of those things, if you are eating a healthy diet, you will get those from what you are eating. If you are on the pizza and french fry diet, you may not be getting all of those trace elements. Diet is important. All right, so basic chem stuff. Very quickly, we know we've got the protons and the neutrons in the nucleus. So our protons are going to make up the atomic number, giving the identity to the element. And then together the protons and the neutron make up the atomic mass. Electrons are so tiny and so small, it would take thousands and thousands of them to add one atomic mass unit. And the atom cannot hold that many electrons, so therefore we basically discount the mass of the electrons. If we lose an electron, then we are going to become positive because we will have more protons than electrons. And if you are a positive ion, then you are a cation. If you are gaining electrons, then you have more electrons than protons, so you are negatively charged. So then you would be an anion. So the way that we can easily remember that cations are positive is because in the name cation, the T is a plus. So if you write it out, cat, ion, plus in the middle, it's positive. And that is a cation, not a cation. I know we're in the South, but it's a cation. All right, we can also change the element by adding extra neutrons, and that is going to give us an isotope. Generally, isotopes don't do anything except change the atomic mass, but sometimes if you have an uneven balance of protons and neutrons, then they become unstable and they will break down, giving off radiation. And the rate of decay is known as the half-life, and half-life calculations is all based on the ratio of the number of the original isotope and its stable product. And then you can figure out how old something is based on that. When we do our evolution unit, we will talk about half-life calculations then. But radioactive isotopes can be very helpful. They have been used many times to make breakthrough discoveries in science about DNA. We'll talk about those in our DNA unit. But basically, you can use radioactive isotopes such as phosphorus, which is part of DNA, and then that is included in the building of new DNA, so you can trace where the DNA goes based on the radioactive signature of the phosphorus. So here they are cultivating some cells, and they give them a radioactive tracer, probably phosphorus, and then that phosphorus is included into the new DNA. As the human cells grow and replicate the DNA, then the radioactive DNA gets passed on, and they can trace that through the different generations, seeing where the radioactive DNA passes. And we'll talk about different applications of that. That can typically be red. You can't just look at it like, oh, it's glowing blue. It can be used by using something called a scintillator. And what happens is the radioactive signature is detected by this machine, and then it creates a graph and they can tell where everything are. Radioactive isotopes are also very important in medicine. You can use it to trace metabolic pathways using what's called a PET scan. And that is where a radioactive molecule is introduced into the body. This is called positron emission tomography. And as the radioactive isotope travels through your body, it gives off positrons, and then they use the PET scanner to trace that, and that can be used to detect things like cancer, heart defects. And this is also used in a CAT scan or a CT scan, and that is computerized tomography, and they will inject a radioactive tracer into your blood. If you've ever had a CT scan, they may have put an IV in, and they put in a radioactive iodine, which would then go through your body, and it creates a radioactive signal in your arteries and veins, which gives a contrast to the x-rays that are bombarding you so they can trace different things. Here, so this is cancerous throat tissue that was identified using a PET scan and imagery, and so that is probably thyroid cancer. And one of the things that is interesting about thyroid cancer is the way they treat it is they give the person pills that have radioactive iodine in it. And so as the radioactive iodine is being used to make that T3 and that T4 hormone, it actually destroys the thyroid gland. And so the treatment of thyroid cancer is to completely eliminate the thyroid, and then the person will have to take thyroid supplements for the rest of their life. All right. Okay, electrons in a neutral atom, protons are going to equal electrons. We change that, you get a cation or an anion. And of course, we've all seen the image of here's your nucleus and here's your little rings going around, and those are the electron levels. That's called the planetary model, which was found by Bohr. And as we hopefully know, that is completely incorrect. Electrons don't travel around in nice little circles. They travel in complex patterns called orbitals that have different shapes based on the number of electrons and all of that stuff. But the important thing to know about the energy of electrons is it can change and jump from one energy level to the next. And so as electrons absorb energy, they are going to jump from an inner level to an outer level. And of course, electrons don't like to be excited, so then they return. And then as they return, they will lose the energy that they gained. And that's typically seen as like, this is how fireworks work. You have the electrons around your elements. When the firework explodes, the energy of the explosion excites those electrons, and then those electrons are like, ugh, I don't like it out here. And so then they drop back down to their ground level, and they give off that extra energy as light, which are pretty, and then we ooh and we ah, and we completely ignore the environmental impact of all of the fireworks on everything. Because they're pretty. And why is this important in biology? The main reason that this is important is this is what drives all of life. This is what makes photosynthesis work. So we have electrons that are in water that get removed. They get excited by sunlight to a higher energy state. And then that higher energy state electron is used to build sugar through a complex process. And then that sugar is used to build starches and proteins and all this other stuff, and to run cell respiration, which gives us the energy to live. So everything is about these excited electrons and using those excited electrons to drive all of our cellular processes. So it's critically important for the functioning of life. All right, so when we talk about electrons, we talk about the valence electrons. So those are the ones in the outermost level. And for the most part, happy atoms have 8 electrons, or the octet rule. There are notable exceptions, hydrogen and helium, because they only have 2 electrons in their shell. And other ones that are notable are things like phosphorus and sulfur. They are different. They incorporate different orbital levels other than just the valence shell. So phosphorus likes to be pentavalent, meaning it likes to have 10 electrons, and sulfur likes to be hexavalent, having 12 electrons for a complete shell. But this is the basic of all chemical reactions. So your basic bonds, your ionic and your covalent. So in your ionic bond, they are going to give and exchange electrons. One becomes positive, one becomes negative, and then they are sucked together by an electrostatic force. And they are going to typically be solid at room temperature. They typically will dissolve in water unless the ionic bond is stronger than the ability of water to pull it apart. If they do dissolve, then we have what is known as an electrolyte, and that is because electrolytes can conduct electricity. So salt water conducts electricity. And this is critical in biology for the conduction of your nervous system and muscle contraction. And this is why sports drinks have electrolytes in them, because as you sweat, then you lose lots of salt in your sweat, and then you don't have enough salt to conduct your nerve signals, which tell your muscles to contract and to relax. And then you need those ions from the salt for proper muscle contraction. So if you don't have that, that can lead to cramping. But a lot of cramping is simply due to the muscles being tired, not necessarily due to a lack of electrolytes. But basically, Powerade, Gatorade, that is Kool-Aid with salt in it. So you can make that a whole lot cheaper for yourself. All right, so here is your sodium with 1 valence electron here, and here's your chlorine with 7. So sodium loses, then this becomes positive, this becomes negative. They're then drawn together because the opposite charges attract one another, and you get table salt, which is great because chlorine is a poisonous gas that if you inhale it will kill you. That's the active ingredient in mustard gas. And sodium at its elemental state is a metal that you can cut with a butter knife, and if you throw it in water, it will catch on fire. So burning metal and poisonous gas equals salty flavored goodness that you need to live. Chemistry is cool. All right, we also have covalent bonds. This is where the atoms are sharing electrons. So you can have single bonds, double bonds, triple bonds, but essentially neither of the atoms wants to give up their electrons. So they are going to pass the electrons back and forth between each other. That sounds like, oh, they are being nice and they're sharing. Actually, they're being super selfish and they're not allowing the other one to take it. Let's not get into moral discussions about electron behavior. All right, but if there is equal sharing, then each atom has the same charge. So we say that it is a non-polar covalent bond. If one atom takes the electrons a little bit more than the other, then the electron hog will have a negative charge, and the electron non-hog will be slightly positive, and that's what it'd be called polar, because you have one side of the bond is slightly positive and one is slightly negative. That's huge, huge, huge polarity, non-polarity in biology. That's going to be critical for understanding how our cells work, especially when we talk about the membrane and transport and all that stuff, because it results in all of these different intermolecular forces. When we talk about things dissolving in water, Ions are together and they get pulled apart by the water so that they are then surrounded by the water, which we'll look at when we do the water lecture. But covalent bonds are actually stronger than ionic bonds. So water cannot dissolve a covalent bond. So when something like sugar dissolves, the sugar molecule is not breaking apart. The water is separating the individual sugar molecules from one another. And the individual sugar molecules are held together by these intermolecular forces, or forces between molecules. And those bonds between molecules are actually pretty weak. But the internal covalent bond of the molecule is actually very strong. All right, so this is two atoms. These are the electron clouds. And then they blend together, and that's your bond in between there. You can also have, this would be a single bond sharing 2. You can have a double bond sharing 4. Or you can have a triple bond where they're sharing 6. That's what happens in between two nitrogen atoms. And the more electrons that are shared, the stronger the bond. Here we have covalent bonding in several important molecules. Methane, we see that we have 4 bonds of carbon and hydrogen, and that is going to evenly distribute around the molecule. Carbon and hydrogen bonds are nonpolar, so this is going to be a nonpolar molecule, which is why it is going to be a gas at room temperature, because there are no intermolecular forces holding it together. Whereas water, the way that water operates, the oxygen is the electron hog. It has a higher electronegativity. That is simply how much it wants to gain the electrons. So because oxygen has an electronegativity of, I want to say 3.5, and hydrogen has an electronegativity of, I think, 2, it causes the electrons to be drawn towards the oxygen at a higher rate. So because the oxygen is sucking in those electrons more often, then we get a more negative charge here, making it slightly negative. And then there's less electrons around the hydrogens, so it is slightly positive. And so what we see in this, because the molecule is an asymmetrical molecule, we can split the water molecule so that it has a distinct slightly negative side and a distinct slightly positive side. And that makes it a polar molecule. And that's what allows water to stick to other water molecules and give it all of its properties, which is going to be in the next lecture. Other types of bonds, you've got the hydrogen bonds. So if you have a water molecule, and this is slightly positive, and this is slightly negative, and you've got another water molecule, then this slightly positive will be attracted to that slightly negative. That is an intermolecular force. That is a hydrogen bond. And hydrogen bonds are also part of what is known as a van der Waals force, or a London dispersion force. And these are just random movements of electrons that allow for molecules that wouldn't seem to bond together. So this is like Velcro. Velcro is not a permanent bond, and the more Velcro interaction you have, the stronger the bond is. So if you have just a tiny piece of Velcro, one intermolecular bond, it's going to be very, very weak. But if you have a giant surface area of an intermolecular bond, like a fat molecule, it may be strong enough to help hold it together. So the accumulation of all these weak bonds can lead to a stronger bond, but it's still not as strong as an individual covalent bond or an ionic bond. But they are important for helping with different properties, and that will come into play when we talk about fats and oils and the structure of the cell membrane.